Good morning, everybody. Thank you for being with us this morning. I'm Elisabeth Padolka from Belgium and Christian Schonin from Denmark, and it's our pleasure to start hepatitis virus session. You will see that it's not necessary to have a word hepatitis with a letter after that to cause one, and it's my pleasure that my co-chair will introduce the first keynote speaker.
And our keynote speaker this morning is Professor Albert Bosch. He is a professor of microbiology and head of the enteric virus laboratory of the University of Barcelona. His research spans both basic and applied aspects of all enteroviruses, and his topic today is on hepatitis A virus evolution and its implications in outbreaks.
Thank you. Thank you for the introduction. And first of all, my appreciation for the organizers of this terrific conference for inviting me to give this lecture. It's a real pleasure to be here. I'll talk about hepatitis A. And hepatitis A, it's actually known from all time. Already Hippocrates mentioned jaundice in his De Morbis Internis. Later on, he mentioned that when jaundice was accompanied by diarrhea, it was a somewhat more benign form of hepatitis. And it was a Scottish scientist, George Clagon, that really described infectious hepatitis in Minorca. Hepatitis, I'll give some background on a virus that I believe it's a very peculiar virus. It's a Picornaviridae, so one of the best known families in virology. And despite an efficient vaccination, still there are every year around 47 million cases, with 14 million of them that are foodborne and 14,000 casualties. It's an efficient vaccination, and because there is a single serotype. But despite this stability, this antigenic stability, the virus is able to multiply, producing quasi-species, so following a quasi-species distribution. So it means that at the genomic level, it's able to vary as much as any RNA virus, but at the protein level, it stays very stable. It's still causing huge outbreaks, like the one in 2017 in the US, that was in this notorious hit parade ranking number one. And as you can see here, it was a very special outbreak. And I'd like to point that the normal mortality associated with hepatitis A is 3 per thousand, 0.3%. And in this outbreak, deaths were 1.2%, accounted for 1.2%. Hospitalizations were around 60%. In some cases, in states like California or Michigan, look at this 3% mortality. So it was an unusually high mortality compared to what's expected. And the problem is still going in the States and everywhere. I mean, we have outbreaks going on all the time. Now, this is one of the last that I could recover, like for instance in Florida, and look at the number of cases in 2019. It's 4 times the ones that were reported in 2018. So really, it's something that is growing in the US and everywhere. People at risk, well, no surprises here. People over 60, people with any underlying chronic disease, chronic liver disease, people immunocompromised, people that have that are homeless, unstable housing, drug user, inmates, so nothing new under the sun in this case. Which is the problem with hepatitis A? Well, after exposure, symptoms usually correlate with transaminase peak here, and this liver inflammation is reflected by this transaminase peak. Diagnostics is done through IgM, and there's a lifelong lasting IgG presence. But the problem is that if there are symptoms like jaundice that There are usually it's only in adults and never in children below 6 as a rule. But viremia precedes the symptoms, the onset of symptoms, and virus excreted in feces is also preceding symptoms and lasting after the end of the symptoms. So there's no way to prevent the spread of the virus if you don't prevent actually the infection. It may be transmitted foodborne, as I mentioned, and an infected host can probably shed about 10 to the 8th per gram of feces, and somewhat these vehicles, water, bivalves, berries, etc., several risky matrices or fomites can contribute to the spread of this virus. For this, there are several factors important here, and one of them is virus stability, together with the amounts of virus shed, the degree of viral contamination, if there is any treatment, any processing of foods, treatment of water, likelihood of infection after ingestion of a few particles, or susceptibility of the host. But the key factor here is the virus is stable enough to keep the infectivity when it's out of the body, when it's excreted out of the body. And hepatitis A is a very robust virus. Here you have a very old study that we performed with several viruses, and this is when viruses are suspended in PBS and when viruses are suspended in fecal suspensions. And just this is the curve of hepatitis A virus survival, and this is the curve of poliovirus survival. So there is really a big difference among two viruses that are very close to each other in terms of phylogeny. Okay? And what's behind this? Well, let me tell you something about Hep A. I'll give some background information because I believe it's good so that you can follow what I'll tell you later. We have a very slow-growing virus. Anybody trying to culture hepatitis A suffers from that. And the reasons for this being so slow is a highly inefficient type 3 internal ribosome entry site, IRIS. And the virus is unable to shut down cellular protein synthesis. So it has to compete with the cellular machinery for protein synthesis. And at the same time, this causes that it has a highly de-optimized codon usage. What do I mean with this? So it means that in order to avoid competing for the tRNAs employed by the cell that's still synthesizing proteins, the virus employs what we call rare codons. Rare codons are defined as those whose frequencies are less than 30% of that of its most abundant syndrome in each of the codon usage tables. And hepatitis A, compared to, for instance, other picornaviruses, has a lot more codon families with rare codons, and the total number of rare codons is also significantly higher. And this has something to do with the robustness of the particle, of the virion. Here you have the poliovirus translation that is according to the cellular codon usage. Okay? But this is not a problem for the virus because this is depleted. HIV translation has employing these rare codons strategically located that slow down the pace of the elongation. And in doing so, the protein get a proper folding, and the particle is more stable, is more cohesive. Okay? So we have a more robust and cohesive particle due to this. When we artificially shut cell protein synthesis down with actinomycin D, for instance, then we somewhat convert the hepatitis A virus into polio-like in terms of growth virus. Okay? This would be our hepatitis A with these rare codons, and when we artificially shut down protein synthesis, we convert this virus in a somewhat polio-like virus, a much faster replicating virus, but that loses the characteristics of this robustness. Okay, the virus has a lower thermal stability, is less acid stable. Hepatitis A can stand pH 2 easily. In presence of biliary cells is a hepatotropic virus, can stand biliary cells. This new guy cannot. And in the presence of high pressure, the virus suffers this decay, this fast virus, and this is the parental strain. Okay? So we have a fast virus, but a much weaker virus. And incidentally, this is a really long story, but just to see that by this approach, by adapting the virus to grow in the presence of actinomycin D, so in conditions of cellular shutoff, we finally achieve to have a fast-growing strain. So this is the plaques of the parental strain. These are the plaques of this fast-growing strain, so that they look very much like polio plaques. And actually, this adaptation to grow in conditions of artificial protein shutoff, it has a ripple effect because mutations in the iris, in the internal ribosome entry site, produce that the iris becomes a fast iris. We have an optimization of the codon usage, and then we have a lot more particle formation. But these particles are weaker than these particles, are less robust, but that antigenically identical. Okay, let me tell you now something about the phylogeny of the virus. We always said, one of the first sentences in my papers were, okay, it's a countdown, all right. One of my first sentences was hepatovirus. Well, hepatitis A is a prototype of the hepatovirus, and is the only member of this genus. Then all of a sudden, a seal virus popped up, and it was termed FOPI virus. But this was just the beginning, and since the isolation of this new virus, new hepatoviruses appeared, affecting bats, rodents, hedgehogs, and shrew. Then marsupials, and I can continue with fish, etc. So we have found hepatoviruses or hepatovirus-like viruses in many hosts. And this points to which could be the origin of this virus, from where it has evolved to the present virus. This you have the phylogeny in human up here, and you can see here shrew, squirrels, seals, bats, of course, rodents, hedgehog, rodents, etc. And here you have these evolutionary relationships according to the host or according to the diet. And you can see that they can be grouped here, these hosts, hedgehog, shrew, bats, as insectivorous. And this pointed the idea that maybe there could be an ancestor of hepatitis A that could be an insect virus. And the truth is that when the crystallography of hepatitis A was resolved, and this is only about 4 years ago, it was very hard because of this slow growth of the virus, it was seen that it differed with the characteristics of polio, for instance, or foot and mouth disease virus, with other picornaviruses, in, for instance, having a very smooth surface without canyon, and with a loop here that is very characteristic of cricket paralysis virus, so actually an insect virus. So once again, it rang a bell that, maybe the ancestor of hepatitis A could be in an insect. Okay. Trying to investigate a little bit more on that, and with our experience on the codon usage of hepatitis A, we build this distance matrix for similarities in codon usage between a series of viruses and hosts, having in mind that the usual thing is that viruses and hosts, the viruses adapt to the host codon usage. Okay? And in this matrix, you can see that dark blue represents maximum similarity, and dark red minimum similarity. And here you have hepatitis A virus, HIV, and here you have other viruses or hosts. And what you can see here, there's a big homology among these guys here. But getting getting down to what happens with hepatitis A, the most similarities were found with cricket paralysis virus that I mentioned earlier, with Drosophila C virus, and with Cryptosporidium virus. Okay? So these are the viruses that have a codon usage more similar to that of hepatitis A. And when going to eukaryotic host, Cryptosporidium parvum, Schistosoma manzoni, so a platyhelminth, and Brugia malayi, a nematode, are the ones that are closer to hepatitis A virus regarding codon usage. So it is tempting to say that the ancestor of hepatitis A probably originated in one of these viruses. The closer would be actually cricket paralysis virus or Cryptosporidium virus, and then through host switch, probably it got to the viruses that we have now associated with hepatitis A. Well, I'm here, and this is a clinical virology meeting, and I'm here to talk about outbreaks and about how these factors affect hepatitis A evolution and variability in outbreaks. In developed countries, despite this vaccine that is really effective, we still have foodborne outbreaks, and we have outbreaks that affect the men having sex with men community. In a much lesser extent, it's travel-related cases. This is almost now negligible. And the problem is also the situation. The situation here is, in this graph, in this figure, you can see child immunity rate. So prevalence of hepatitis A. Hepatitis A is endemic in these areas. This means that infection is acquired in childhood, providing a lifelong immunity. And here is susceptibility, adult susceptibility. And it's just the opposite, of course. And what happens is that through global food trade, we have food produced in these areas that it's exported into and imported in these areas. Okay? So this is one of the problems with foodborne infections. We have now tools to control a little bit these infections. There is an ISO for the quantitative and qualitative detection of hepatitis A as well as noroviruses in risky food matrices. We have multiplex systems, we have even commercial kits, so this should not be a major problem. And obviously, positive samples should be characterized, should be typed. And for this, the most universal region is a VP1-2A junction that enables to determine genotypes and subgenotypes. Genotypes that differ each other in a 30% difference in nucleotide, and subgenotypes that differ in 10% from each other. And geographically, we have this distribution of subgenotypes. 1A and 1B, as you can see, are the two most prevalent subgenotypes that are basically everywhere. Subgenotype 1C is common in Bolivia and in Peru and has caused problems with food in Europe. And subgenotype 3A here is from the Indian subcontinent, but is now causing problems in Europe as well. Here you have some examples of outbreaks with the type of food involved, the country of origin, where it has been detected, and the subgenotype associated. Okay? This could be obviously much, much longer. It's just an example of the situation. Okay? And let me say something about 3A. 3A is, as I said, is a subgenotype that's common in India, but that has been expanding to Europe, to Russia, and also to Asia, including Japan. Usually it's not associated with foodborne hepatitis transmission, but here in Copenhagen is one of the exceptions that we had here cases. And the concern here is whether it is associated with fulminant hepatitis. Fulminant hepatitis is the most severe outcome of hepatitis A, And there is a controversy. In Japan, there is a no genotype association together with a genotype 3A association. So in France, they claim that fulminant hepatitis is caused by a genotype other than 1A. In India, no genotype association, and US genotype 1B. In our case, we did a meta-analysis of all our data, and what we found is that 1A accounts for 66% of the cases and for 30% of fulminant hepatitis. 1B accounts for 14% of all cases and 30% of fulminant hepatitis, and 3A accounts for 21% of cases and 41% of fulminant hepatitis. So being plain safe, I would say that genotypes 1B and 3A could be mostly associated with fulminant hepatitis, and particularly 3A in our case. In Korea, for instance, it was shown that the disease is more severe when it is caused by 3A. So hospitalization, 20 days instead of 7A for 1A. 63% means that it's a prevalent subgenotype. And also you can see, according to transaminases and bilirubin, that levels are higher than for instance, 1A. So it's more severe disease in any case. At home, what we found is that I mentioned earlier that below 6 it's difficult to see symptoms in children of hepatitis A. In this case, with 3A, we saw symptomatic hepatitis in children below 3. And for instance, what we found is that first accounted altogether at 27% compared to 55 for 1A or 18% 1B, but the mean age of the patients, look at that, is 3.6 years. And I'm talking about symptomatic cases. So 3A causes symptomatology in children below 6, which is also very unusual. Okay, now let's do I'll talk about some outbreaks and what we learned from these outbreaks. We usually have outbreaks like this one. It was a berry outbreak, a strawberry-caused outbreak. As usual in the EU, nobody knows where it came from, probably from Poland, although some products were originated in Italy as well. Italy was where the maximum number of cases were reported, but it was, as you can see, a global in Europe, but not in the Spanish Peninsula. And why was this? So we didn't see any cases, well, none in the Balkans, but I don't know anything about the Balkans, but it could be a lack of notification, no imports, or the immune status of the population. The truth is that there is a notification, so it's not a problem of not notifying the cases. We don't have imports of strawberries. The truth is that Spain is the first producer in Europe and actually the second in the world, so we hardly have any imports of strawberries. We have berry mixes and things like this that come frozen from China, but not in this case. We don't consume raspberries, blueberries. It's not traditionally it's not consumed, and the population older than 60 are naturally immune. I'm sorry to say that I'm immune, and the vaccinated group at this time included those from 20 to 27 years old. Okay, so vaccination and all these cases probably made that we didn't have cases. It's very important to type and to have the characterization of the positive isolates. And let me show you some examples. For instance, we had in Catalonia a men having sex with men outbreak that affected 185 cases, and we had two casualties. One of these patients was a baker, and he was handling food, and he caused a foodborne outbreak with 21 cases. Typing was relevant because this happened in a small town, and tap water was blamed for this outbreak originally. Okay? And thanks to the sequence, we could trace that it belonged to the MSM group, and that actually this guy was an HIV positive person, and he was under antiretroviral therapy, and the physician confused the secondary effects of the therapy with the symptoms of the hepatitis. Okay, so that's why. But anyway, and he did an improper food handling, which. This was it's another example of the importance of having genotype or subgenotype information. We had two concomitant huge outbreaks together. One was in Valencia in 2008, that was in 2008, with 100 cases. This was economically a disaster. 4,000 tons of clams were immobilized. They were imported frozen from Peru. And there were 3 million euros of losses. A lot of claims for economical compensations, etc. And at the same time, we had men having sex with men outbreak. This corresponds to here. This is the Peruvian outbreak caused by 1C subgenotype, and here is the MSM outbreak that was caused by 1A. So we could really differentiate. Actually, we can differentiate geographic origins. We have clusters of cases from Morocco, from Colombia. We have several clusters with the MSM groups, and in Egypt. So in this way, we can trace geographical origins and sometimes even the route of transmission. And it's very helpful when, for instance, you have this cluster of several outbreaks caused by Peruvian clams, for instance, and here there was these samples from one guy that put a claim that he had eaten these clams, the clams that caused this outbreak. But anyway, now lawyers know genetics, and this guy also didn't get a compensation because this sample belonged to an MSM outbreak. Okay? So he didn't get the compensation. In this case, we had an outbreak in Europe. It was caused by 3 strains. One was described in in London. These are the flags of the cities. In London, although it originated in the UK, in the Canary Islands, or in the Canary Islands, this case. This one was reported in Amsterdam, but it was originated in the UK. We had some samples as well in Spain, some cases. And this one originated in Berlin, and it was reported in Berlin. But we also had cases. So we had these 3, with these 3 strains circulating in the MSM community, and we could find the 3 strains in water of this is a natural park, the Llobregat Delta, that receives some tertiary effluents, and at the same time, water from this water is employed for irrigation of artichokes, rice, and lettuce. Okay, so, and these are the data we found up to 10 to the 5th genome copies. We could characterize it that there were the 3 strains. So, obviously, when an outbreak is circulating in the population, you can find in the environment what's excreted, and it's a good indication of what's circulating, sometimes with or without symptoms. Okay. Well, let's get back to rare codons. So now we know that the need to preserve these rare codons may be the reason why there is an antigenic change. So why, despite this evolution at the genomic level, this is not translated in changes in antigenicity, usually. What we did was a quasi-species analysis under the pressure of monoclonal antibodies. These 2 monoclonal antibodies, these 2 monoclonal antibodies, H7C27, K34C8, and these in yellow are the epitopes of these antibodies, and most of the changes here, 86% of changes in VP1 and 68 changes in VP3 were transitions that maintained codon frequency, so that there was no changes at all. you can see that there is hardly any coincidence. This light blue here, for instance, is when changes affect directly an epitope. In white, you have rare codons. So yellow, the antibody epitopes, rare codons, and the coincidence here in blue. Okay? But nevertheless, we found variants. We found variants. This was in an outbreak, and we could see this strain that was unable to be neutralized by the Havrix vaccine serum, by a vaccine serum, by convalescent serum, or by this monoclonal antibody, K4C8. Okay? So this guy was really a escape variant. Okay? And this was originated in an outbreak, and this made us think that maybe what we're doing when we use Sanger sequencing is just seeing the tip of the iceberg, and then we went through trying to study through deep sequencing what happens in an outbreak under immune pressure. So we had to set the following situation. Last year there was a shortage in vaccine production, and hepatitis A vaccine was unavailable. Why it's a little bit silly, but apparently when vaccine producers produce a vaccine like the papilloma vaccine, they reduce the stocks of production of other vaccines. And one of the vaccines that were reduced was hepatitis A. And then there were a huge outbreaks, and this was a global shortage. And every health system in each country decided what to do in this case. In Spain, for instance, what they did was to reduce the number of doses. So an immunocompetent host received a single dose, an adult single dose. And an immunocompromised host received 2 doses. Okay? But Public Health England took other decisions, but all countries had to reconsider their policies. All right? So we had this situation. And in this outbreak, we had vaccinated patients. So we had patients that had received the vaccine, together with patients that did not. Okay? So it was a good possibility to study this. So we conducted this, we prepared with this experiment, amplifying these overlapping fragments covering the entire 5' non-coding region, the complete capsid region, and part of the 2A region here. And what we found is that we found changes, we found mutants in non-vaccinated patients, but also in vaccinated patients. Okay? So these in here, these are we are following always the crystallographic model of hepatitis A. As usual, VP1, VP2 and VP3, blue, green, red. And as before, green are the epitopes, and white, in white you can, sorry, in this case in blue, because in white you can hardly see any. In blue is when we have changes near the epitopes. Near means 1 or 2 nucleotides apart. Okay? Just for instance, these are 2 vaccinated individuals, M5 and M6. This guy was a 34-year-old HIV negative guy that had been vaccinated, and you can see it has, here is a change in the glycophorin A binding site. It has this change in the putative receptor binding site, and it has this change. This is another guy, also vaccinated, and it has this change in the immunodominant in VP1 site here. Okay? And what's more interesting to me is when you look at the non-synonymous mutations around these epitopes that I mentioned now, the VP1 immunodominant site, the glycophorin A binding site, and the putative receptor binding site, you can see that there are no differences between non-vaccinated and vaccinated people in the percent of non-synonymous mutations, but there are differences when you look at the percent of haplotypes bearing non-synonymous mutations, okay, you can see that there is a clear difference between what happens in the immunodominant site in vaccinated and in non-vaccinated individuals, in the same way as in the glycophorin A binding site. There are no differences in the receptor binding site. Okay? So this means that there are mutations that are fixed, that are prone to be fixed in vaccinated individuals, at least in these conditions that we had of vaccine shortage, that leads to the potential arise of variants that could escape to these vaccines. Okay? And I will provide here some of my take-home messages. We are in front of very, very peculiar ancestral virus of unknown origin. I didn't mention peculiarities of hepatitis A that fascinate me, like for instance that it is a virus that can have a dual morphology of being a non-enveloped virus, as we always thought, and an enveloped virus, and is produced from the hepatocyte as an enveloped virus. Okay? But I didn't mention that, but just for you to know that it's really a strange guy. It has a unique codon usage that determines this virion robustness and antigenic stability. This is a consequence of this impossibility to shut down protein synthesis. Despite efficient vaccines, they are really efficient. They are expensive, but they are really efficient. Hepatitis A is re-emerging in developed countries. It is. And maybe the more controversial part is that suboptimal vaccination may lead to the fixation of mutation in antigenic sites and maybe to the appearance of vaccine escape variants. Okay? And vaccination policies should provide adequate number of doses, up-to-date vaccination schedules, and careful evaluation of prophylactic immunization of contacts, especially when direct to the MSM group and or in situations of vaccine shortage. For instance, it's a well-known practice to conduct a post-exposure immunoprophylaxis in terms of vaccinating contacts. Okay? We've seen that if the virus is already growing, is already infecting, it could not be good to vaccinate this person. This maybe I shouldn't say that, but I mean that maybe the process of the disease will be shortened, but also the possibility to develop hepatitis A virus variants is higher then. Okay, and then I will finish my presentation acknowledging the people that actually did the work. This here is Rosa Pinto. Rosa Pinto is the guru of molecular biology in hepatitis A, I believe, in Europe. He is the one that saw that the codon usage of hepatitis A is very special, and from that built up this story. This is Susana Guix here, that was very active also in this project, as well as Montse de Castellarnau, Aurora Sabrià, I can see here, Aurora Sabrià, and Cristina Fuentes. Thank you very much for your attention.